So hello everyone, I think we can start. First of all, welcome to our first Kusai Neuroscience Festival. Thank you for being here and attending. I want to start by introducing myself. I'm Ahmet Can Sezen, a first year medical student at Koç University. This is my second year at Kusai as a board member, and this year I'm also working as the coordinator of the Neuroscience Festival. We have a great team consisting of our presidents, board members, and active members. We have worked for months for this festival, and I thank them all for their amazing efforts. Before moving on, let me share my presentation as well. I'd like to talk about Kusain a little bit. Here you can see our beautiful family. As Kusain, Koç University Students Interest Group in Neurology, we are a chapter of American Academy of Neurology. But we have a small difference from other science chapter, and which is that we don't have only medical students at our club, but also students from engineering, psychology, and many different majors. This gives us an advantage to see different perspectives and work interdisciplinary, as you will see in the sessions of our festival. Throughout the year, we have organized many different events, and today we are here with one of the biggest ones. I hope it will be a festival in which you have a lot of fun and learn many new things. Now, I will leave the floor to Melike Kocahasan, who is my co-coordinator of this event, to introduce herself and talk about the festival schedule. Thank you.
Thank you, Ahmetcan. Hello, everyone. Firstly, I also would like to say welcome to all of you. Welcome to our first Kusai Neuroscience Festival. I am Melike Kocahasan. I'm a first-year medical student at Koç University, and this is my second year at Kusai as a board member. And this year, I have been working as a coordinator of Neuroscience Festival too. And now I would like to talk about the festival schedule. Here you can see the whole program for 3 days. We wanted to bring together many valuable professors from many different subfields to make this event as interdisciplinary as possible, because neuroscience is so. And we try to find interesting topics for each of these sessions. We hope you like them all. In addition to this, I have a few announcements about the festival. Participants who join at least 7 sessions will receive a certificate, and then we will have a gift drop among those who receive the certificate. Now let me start with today's schedule. Firstly, we will be with Associate Professor Atay Vural to learn the basics of neuroscience. Then Associate Professor Alba Tuninetti will give her speech about language in the brain, and today's sessions will be completed. So thank you for being here, and I hope all of you find this festival beneficial and you enjoy it. Okay, now I would like to give floor to our presidents, Orhan and Leay. Orhan, we can start with you if you want.
Okay. Hello, everyone. It's a pleasure to see such an amazing event. I'm Orhan Talha Özgün. I'm 5th year medical student. I'm very happy to be a co-president of this wonderful social club. I want to thank firstly Ahmet Can Sezen and Melike Kocahasan and all of our other members which contribute to this festival. They made an excellent job. I hope you will enjoy and learn lots of things that make your vision wider. See you at our panels and conferences.
Okay, hello everybody, it's Leay. I'm the other president. And first of all, I would like to introduce myself very briefly. I'm a last year student of psychology and molecular biology genetics, and I am hoping to graduate this year. And then I would like to thank my beautiful team, because these people are so hardworking and incredibly great at any crisis management, and I'm so proud of them for putting through with this and organizing everything, inviting you guys and meeting you guys. It's just a great pleasure. So I'm so happy to see you all. And I hope that you learn a lot of things from this festival, because it is a very tight schedule, but it is packed in a way that you can learn a lot. So have fun, you guys. Thank you very much.
And thank you, Orhan and Leal. Now, we can actually start with our first session. I will be moderating it. Associate Professor Atay Vural is here with us. I want to say welcome to him. And firstly, I would like to introduce him briefly. He earned his MD and PhD from Hacettepe University after completing the MD-PhD combined program. He used intravital and digital imaging techniques to study brain microcirculation in mice for his PhD thesis. During his neurology residency at the same university hospital, he participated in research projects on clinical neurology and neuroimaging. Later, he worked as a postdoctoral research fellow at Ludwig Maximilian University Institute of Clinical Neuroimmunology in Munich, with scholarships from Alexander von Humboldt Foundation, European Academy of Neurology, and TÜBİTAK. His research interests include neuroimmunology, neurogenetics, and neurotechnology. His research is focused on multiple sclerosis, autoantibody-mediated nervous system disorders, and rare neurogenetic disorders, including motor neuron disorders and movement disorders. Now, I would like to give floor to Atay Vural for his presentation. Welcome.
Hello, everyone. Thank you very much for the introduction. First, I would like to thank Hüseyin and especially the directors and organizers of this first Neuroscience Festival event. It's really a pleasure for me to be the opening speaker of this event. I hope in the following years we will see the following of these nice events with increasing number of participants and professors. So thank you. I really think this group is really important. As Ahmet Can mentioned, it's unique in terms of the members. So not only the medical students are involved, but it's a truly interdisciplinary group. And it's I really like this because the nature of neuroscience is interdisciplinary, as I will talk about soon. So I think this is really a nice group and it's a nice event. So I will share my screen now. I think you can see it now.
Yes, we can.
So the title is Neuroscience 101. So it will be a general introductory lecture for neuroscience, a little bit history, and more on the today and near future of neuroscience and its interdisciplinary nature. So what are the fundamental questions of neuroscience? As you all know, it's mainly dealing with two questions actually. So how do we think, or how do the human brain function? And the second major question is what are the bases of diseases of the brain and how we can treat them. So regarding the human brain function, the major questions are: what is the biological basis of learning, memory, behavior, perception, and consciousness? And what makes the human brain unique other than the other organisms? How do the billions of individual components of the brain work together to generate a behavior? So the major aims of neuroscience are to try to answer these questions. As you all know, one of the primary principles of neuroscience is the neuron doctrine. So for today, it's a very basic and simple concept, but its discovery was a huge thing more than 100 years ago, and it started neuroscience actually. So I will start my lecture with this process first, because it has many things related to the nature of science in general. So now we know that the neurons are individual building blocks of the brain and nervous system, and they are separated structures which are connected by synapses, and their specific connections in terms of network and their specific firing patterns make up the networks, and the function of these networks make up the brain, the behavior. But it was not so easy in the beginning to discover that. So in the 19th century, in the beginning, the staining methods, histological staining methods available could only show us a very low resolution images of the brain. So it's from a textbook of Ranier's, and these are the neurons. So at that time, scientists thought that neurons are made up of cell bodies and some small neurites, extensions of the neurons. At around 1870s, there was a huge technological development for that time. Camillo Golgi, an Italian scientist, discovered a specific staining method named as silver staining. And by using this method, he could see high resolution images of human brain, the neuronal cell bodies, the long neurites, dendrites, and axons. But of course, staining method is not enough. Microscopes are necessary, and at that time, similar times, technological advancements, like just now, I will show you soon, were happening. Some first microtomes were developed, and some nice microscopes for that time, advanced microscopes of that time were being developed, and Golgi had access to these microscopes as well, and he could make the first observations.
Atay Hocam, sorry to interrupt, but we are still seeing the first slides. I guess there's a problem with the screen sharing. Can you do it again? Okay, now it's correct.
Okay, thank you for saying that. Not too late because so of course
yes, I didn't want to interrupt, but
no, so I witnessed that before, and it was much later. So thank you.
Okay, can you also do it full screen?
So I'm changing slides. Can
Thank
you see
you.
it? I'm
Yes,
changing
we
slides,
can see
please.
it now.
Okay,
Thank
so
you.
I talked about the neuron doctrine. So as we know, neurons are individual building blocks. So this is the textbook from 1870. The neurons, only the cell body and as short neurites. But Golgi discovered the silver stain and used the fancy microscopes of that time, the high technology microscopes of that time. And he saw such images. So this is one of the actual sections that Golgi stained, and of course it was imaged later, recently, to reflect on what he saw. So this is one of his slides. So he saw this image, and by seeing this image, he thought that these neurites, which are supposed to be dendrites now, are nourishing the cell bodies, and the extensions, neurites coming from these neurons, are connected all together. And he thought that these are not single individual cells or neurons, but this is a syncytium or a network or a reticulum. And the basis of brain function is a reticulum. So this was called at that time the reticular theory. So the scientists, Golgi and other scientists before him, thought that brain functions as a syncytium of these structures. Of course, we know that this is completely wrong now, but how it was discovered that it was wrong? Thanks to the great man Cajal, Ramon Cajal. So he's a Spanish scientist. He was younger at that time, in his 20s. He was an anatomist. One day, he went to visit one of his friends in another city of Spain, and his friend just came back to Spain after working with Golgi to learn his newest staining method. And in this lab, Cajal saw the silver stains brain slides for the first time, and he was like, he thought it's like a magic. So he was like, okay, I should work on brain from now on. So this is one of his textbooks, and he says, a look was enough. Dumbfounded, I could not take my eyes from the microscope. And he started working on brain. So he used the similar staining methods. He developed them even further, and he developed or used additional staining methods as well. He also had access to the modern microscopes of that time, and he devoted his life to brain histology and anatomy. And what he developed was the neuron theory. So unlike Golgi and his precedents, he thought that neurons are single cellular structures, and they are not in continuum, or they do not form a reticulum, but they are connected to each other by very thin spaces, and they talk to each other through these very thin spaces. So it is called as the neuron theory, unlike the reticular theory of Golgi. Of course, now we know that the neuron theory is correct. And in addition, he also saw some very tiny spikes on top of the neurites after silver staining. He thought that these are important structures for communication between neurons. However, Golgi, although he also observed the same structures, these tiny spikes, he thought that this is a technical artifact. They are not real, but they are just technical artifacts. But Cajal was really an excellent observer and scientist, and he just didn't think these are artifacts. He stained them by using additional staining methods based on silver staining again. He observed the same structures again, but this was not enough because they were also based on silver staining. What he did was he used completely different staining methods in addition, and he observed these again. And indeed, these are now very well known dendritic spines, the synapses between neurons. And indeed, this was very important structures for neurons as well. So Cajal was really an excellent scientist. He was not blindsided with dogmas or the thoughts before him, but he was an observationist. He just observed what he saw, he reflected on them, he thought on about them, and he developed novel theories. He was not bound with pre-existing theories. So this is one crucial feature of a scientist, opposing the dogmas. Cajal was not a brilliant scientist only, but he was also an artist. Being a Spanish, he continued the Spanish école of being excellent painters, and his paintings, depictions of the brain slides he stained, these are his own drawings, are now considered as art material as well, and they are exhibited in several museums throughout the world. Sherrington, one of his colleagues in UK, was also thinking similar with the Cajal, and he was supporting the neuron theory. He developed this further. He thought that the synaptic structures are important, and Sir Sherrington gave the name of synapses at that time. First, he proposed the name syndesm, but after talking with some linguistic scientists or linguistics, he decided on the name of synapse that we use nowadays. What proved actually Cajal was right, of course, is again technology. In 1950s, after the development of electron microscopy, we could see the actual synapses directly. So this proved the theory of Ramon Cajal, because now these are the spaces between two different neurons. So this proves that the neurons are not physically connected, but there is a tiny space between them, and they talk through these synapses. And this proved that Cajal was completely right. So Ramon Cajal and Golgi shared the Nobel Prize in 1906, because although Golgi was completely wrong to interpret what he saw, because he was heavily influenced by the previous theories before him, he was wrong in terms of science, but his methods were crucial to advance science. So for that reason, Golgi and Cajal shared the Nobel Prize in 1906, and especially Ramon Cajal is regarded as the founders of neuroscience now. So today we know that neuron is the building block of the central nervous system, And when we think of a prototypical neuron, we think of soma and dendrites and the axons. So these depictions are from textbooks, and they're actually quite misleading, because most of the time we see single neurons lying neatly. So there are large spaces between neurons. Actually, this is not the case. The nervous system is very complex. It's the most complex structure in the whole universe, and it's very densely packed. There are billions of neurons and trillions, more than trillions of synapses between them. So it's really a chaotic, very complex structure. And we depict synapses like this, but of course, again, it's not a bistructural complex, but there are also astrocytes contributing to synapses, and there are many more synapses around. So these depictions, as always, are actually also misleading. Today, we know that we can classify neurons in terms of their morphology to several different types, but morphological classifications are very subtle or very superficial for us to understand the individual contribution of neurons. So classification is important for science, as Darwin first classified species, and also taxonomy classification is always very important to understand nature better. And until recently, the only way to classify the neurons or other brain cell types was the morphological classifications, but this isn't enough to understand the complex brain functions. And nowadays, we use much more complex molecular-based methods to understand the complexity of brain neurons and other cell types. I will talk about these soon. For example, we know that the 6 layers of the cortex in different anatomical regions are formed of different types of neurons, and each of the neurons in these layers make different types of connections with different types of the brain, and this is called the connectome, the connections between different neurons. So this is a short video. I hope you can see it. So this is to show you the brain. This is from the Allen Institute. Please learn the name of this institute because it's one of the leading neuroscience institutes in the world and USA. They are doing many projects. This is one of them. They are just automated electron microscope methods to get ultra-thin sections from only one brain. They are taking more than 100,000 pictures. They are constructing a 3D image, 1 million synaptic connections. Just 1 million. This is an ongoing. Nowadays, the goal is to understand or identify the connections between these neighboring neurons and also the connections between distant neurons. Identify the connectome. So some numbers. In the mouse brain, there are around 100 million cells. And in human brain, it's 180 billion cells. And around half of them are neurons, and half of them are non-neuronal support cells like astrocytes, microglia, oligodendrocytes. So these are incredible numbers, and the connections between them is even much more, of course. So for a long time, until like 5 or 10 years ago, the major focus was the neurons regarding the nervous system, and almost nobody worked on the glial cells. It was very rare. But now, after many decades of work, we know that, okay, neurons are the building blocks of the brain, okay, but especially to understand the diseases, and also normal brain function. It's not enough to study neurons. We must also learn in detail about the supporting cells, astrocytes, microglia, oligodendrocytes, vascular cells, and other cell types. It's not an extra, but it's a must. Without understanding the brain as a whole, as a system, we cannot understand its function and its diseases. Similar to the neurons, we can use some histological markers to label astrocytes. There are like 10 of them, or microglia. But again, the morphological classification based on histological methods are very subtle. They are not enough to understand the incredible complexity of molecular complexity of these cell types, and we need more advanced methods like omics, single cell omics based methods. So these are the classical methods used in neuroscience until recently: histological, physiological, behavioral, biochemical. By using these methods, we study the anatomy and histology, function, especially the electrophysiological function of the neurons, which is crucial to understand brain function, and the behavioral outcome of the network function of the neurons. We should measure behavior by using some specific behavioral tests, and of course, the neurotransmitters or the other chemicals used by neurons is again crucial to understand brain function, and these are the classical methods used in neuroscience which are extremely valuable. So as you can see, these actually are related to different science fields, science branches. So histology is a specific branch of science, physiology, behavioral science, and biochemistry. Each of these are themselves specific scientific branches. And neuroscience was not a separate scientific field itself in the beginning. In the beginning, scientists working in these specific areas, and many more of them, were working on brain. And around 1950s in UK and in USA, scientists from these different fields came together in several places, and especially in UK, of course, they were getting together in a pub, especially this one, the Black Horse, and they decided that this interdisciplinary effort should be named as a separate scientific field, and they founded the first neuroscience association at that time. So British Neuroscience Association is one of the first neuroscience organizations in the world, and just 1 year later in the USA, Society for Neuroscience was also founded, and from now on, neuroscience was regarded as a separate but interdisciplinary scientific field. Like 2-3 years ago, we celebrated the 50th year of the foundation of these two foundations. It's like, although the neuroscience is regarded as being founded up in the beginning of 20th century, the foundations are much younger, and neuroscience is, as you can see, a young field. Many Nobel Prizes in medicine were given to scientists working on brain, starting from Golgi and Cahal. Sherrington was also rewarded. Otto Loewi, Eccles, Hodgkin, and Huxley regarding the electrophysiology of the neurons. The famous Eric Kandel, the editor of the major textbook in neuroscience, was given Nobel Prize in 2000, and also more recent awards are present. So in the beginning, the Nobel Prizes were given to scientists who discovered the characteristics of individual neurons, anatomically, histologically, electrophysiologically, or pharmacologically. But in the later times, it's given to scientists working on the network function of the neurons. Of course, molecular evolution has contributed greatly to neuroscience as well. First sequencing and then omics methods, and in the very last years, more complicated single cell based methods and CRISPR methodologies. GF fluorescent proteins are vital in neuroscience research. It's indispensable. Transgenic animals are indispensable. So these are all very important for the advancement of neuroscience. So 100 years ago, it now seems easier that scientists could do neuroscience, but actually it was not. So the microscopes of that time seems very basic now, but for that time they were very advanced, and it was not easy to find information, to have access to information, knowledge, technology at that time as well. Actually, it's much easier nowadays to find any kind of information or knowledge, or to reach technology, actually, even find some money for science. It's easier, but what is more difficult is to get the right knowledge, right information, and make a right hypothesis, right synthesis from this huge amount of knowledge. It's the challenging thing, and also it's challenging to learn the complicated technology to analyze these new information. So to overcome these barriers, this age now, starting from 2013, is the age of brain. As you know, like 50 to 60 years ago, it was the age of space race. Like 30 years ago, it was the age of human genomics. And now, in the last especially 10, especially 5 years, it's the age of human brain. So the major projects are on understanding the human brain and human brain disorders. So the Brain Initiative, founded by US and the Europe Initiation HPP and the Minds project in Japan, are all major projects. An international Brain Initiative was created 6 to 5 years ago by the participation of many different countries, as you can see. So the budgets are incredible. For the US Brain Initiative project, it's 6.6 billion. For European and Japanese projects, it's around 300 to 700 million dollars. So these huge projects all aim to understand the connectome and the brain cell types of the brain. So it's called brain mapping. What is the aim? To systematically chart brain's connections, catalog its cell types to the highest detail possible, and their physiological properties. How do they fire? So to understand brain is not an easy task, because it has many levels of complexity. So to understand connectomics, we must think of macroscale, mesoscale, and nanoscale. Macroscale examine the communication between regions of the brain in the global level. Mesoscale interrogates neuronal circuitry at the cellular level. And nanoscale assesses individual synaptic contacts. So even the scale is not one single thing. It has many orders of magnitude in terms of size. It's really complex. So the Brain Initiative of USA is made up of actually by the contribution of other or several many large institutes or foundations, like the Allen Institute, Simons Foundation, Genelia Research Campus, Kavli Foundation, and many more. So it's a huge effort. And one of the major projects of this initiative is called Cell Census Network. Census means nüfus sayımı in Turkish. So they are trying to identify the individual cell types of the brain, especially neuronal cell types. 26 teams in US institutions are contributing, and just a few months ago, 17 papers on this issue, for this aim, were published in Nature altogether. And all of these 17 articles were about the motor cortex of mouse brain, which constitutes only the 1% of the whole brain of the mouse. And they examined these neurons in different aspects. I will talk about them briefly now. They are planning to complete the whole mouse brain by 2023, which is next year, and hopefully the human brain like 5 or 6 years later. And one of the major goals is to develop novel technologies that make us, the scientists, to understand the complexity of brain. So technology development, just like in space race, is actually maybe the major goal of these efforts. So what they did, they used molecular single cell based methods. So for the popular method nowadays is single cell RNA sequencing, as well as single cell epigenomics. And they use these methods in addition to special single cell RNA sequencing to identify the different molecular types of neurons in the motor cortex. And they analyzed more than 1.1 million cells just in 3 months and found 56 different clusters or types. They also developed a novel method, which is a fascinating method named as patch-seq, and they took human brain slices from recently deceased humans, and they can make these tissues alive for several weeks, and during that time they put very tiny capillary electrodes inside the individual neurons, give a fluorescent dye to morphologically examine it. In addition, it's a patch clamp method, and they can measure the electrical activity of this individual neuron to identify its characteristics. In addition, after the experiment is over, they can just suck up all the molecules inside this neuron to this capillary tube to analyze in terms of transcriptomics and epigenomics later on. So they did this method makes it possible to detect the electrophysiological features, morphological features, and the molecular features of a single neuronal cell. So this is one of the figures from this article. This is a famous Tisney or UMAP plot. Please, all of you become familiar with such graphs, Tisney or UMAP graphs. So each of the dots are a single neuron from that region, and for each of the cell, they can identify the molecular types, and each of these clusters are similar cells here, and they can find the transcriptomic properties, epigenomic properties, morphological properties, as I mentioned, and the electrophysiological properties of individual neurons and similar neurons forming clusters. So this is really incredible. Again, another figure from the same article. It's possible to detect individual morphological, molecular, and electrophysiological features of a single neuron. As I mentioned, one of the major goals is to identify the connectome. So they also did 2 structural studies as well among these 17 published articles. For that purpose, they use transgenic animals, especially the Krell-Dox system, viral tracing methods, especially AAV virus, in addition to tissue clearing methods, and the microscope named as light sheet fluorescent microscopy to obtain 3D volumetric cellular level resolution images from human or macaque or mouse brains. So please be familiar with these methods. When you inject AAV virus, specifically genetically engineered to label specific neurons in the injected area, if you combine several of them, you can study connectomics thanks to the fluorescent proteins produced by these viruses. So thanks to this method and tissue clearing, we can study connectome of these brain structures. So they also studied and compared the cellular complexity between human and mouse and also macaque brains, and they identified several exciting information. For example, excitatory inhibitory neurons in the cortex is lower in humans, which means inhibitory neurons are more important or more abundant compared to lower structures. What is most exciting for my perspective is all of these data of these billion-dollar projects is freely available for all of you. So you can enter the brain image library or NEMO archive for the multi-omics data or the neurophysiology data integration website, and just download all of these data, which you are, depending on which you are interested in, and you can analyze by again using the freely available methods developed during these studies. Of course, this is not a very easy job, but it's possible if you are interested in it. So this is, for example, the website of NEMO. This contains the molecular, transcriptomic, and epigenomic information from individual neurons of the motor cortex. Okay, so tissue clearing is one of the exciting methods. What does it mean? When you use some specific chemicals, detergents actually, the opaque brain can be become transparent, and transparency means you can acquire volumetric images from whole of the brain by using some microscopes named as light sheet microscopes. Again, these are also it's possible to build them as an open source because the map, for example, the Mesospin website gives all the instructions to build one. Of course, you cannot do it in your home because they are expensive and requires some expertise, but it's actually possible. So this is a video from a macaque brain. It shows the connectome of a specific brain region. This shows us the different scales, the mesoscale, macroscale, and the nanoscale. So you can see a single individual axon originating from a specific region of the brain. You can trace it throughout its length until it reaches to its target in a completely different part of the brain. You can see the many synapses which are seen next to the axon. So it's just one connection between two different neurons. This is just one connection. As I mentioned, there are especially the website of Allen Institute, Allen Brain Map, harbors these information or these data, the patch-sec data, two-photon data, and other data as well, like the mouse brain atlas, the developing mouse brain atlas, glioblastoma atlas, dementia atlas. It's possible to access this data completely freely. This is from another institute, the Geneva campus. They also have their own project on the connectome, named as the Mouselight project, by using the AAV virus. Again, you can access these websites easily. Okay, the understanding the cell types and the connectome is one part of the story because it gives us morphological, molecular, and structural information. But what about the function? What about the function of these networks, connectomes, and their behavioral outcome? How can we study these? We again need some high-tech methods, which they are high-tech, but they are available actually. We can also access them. We can use genetically encoded calcium or voltage indicators to assess the firing patterns of neurons. We can use a microscope named as 2-photon or 3-photon microscope to image freely behaving animals, especially mice. We can use optogenetics and head-mounted microscopes and automated behavior analysis methods to study the network behavior and the associated behavioral output of these networks. So, for example, this is just a treadmill, and this is a mouse with a head-mounted microscope, and this is a virtual environment, virtual reality environment. So it's like a labyrinth, and the mice thinks it's going through a labyrinth. And when you put a microscope through a cranial window of a mice, you can get images like this. So you will see some sparkling dots. These are individual neurons, and each of these neurons have genetically encoded calcium or voltage indicators. This is why they are sparkling under a fluorescent microscope. And by analyzing these images, serial time series images, it's possible to gain insight into the network behavior of neurons. So this is a head-mounted microscope through a cranial window.
How do nerve cells in the brain process information from the senses and drive complex behaviors? This question has fascinated neuroscientists for many years. In the field of neuroethology, researchers use model organisms like the rat to understand biological processes. Their brain is functionally almost identical to the human. In addition, rats perform very well in learning tasks and are well suited to studies of learning and cognition. In the last decade, it has become possible to genetically engineer neurons in animals to produce fluorescence reporters that change their brightness in response to brain activity and then monitor them under a microscope. With this tool at hand, we are getting closer to the central goal of neuroethology, to get a mechanistic understanding of animal behavior at a neural circuit level. For this, we have to combine various research tools, such as behavioral experiments and microscopy of brain circuits. Photon imaging is well suited for imaging in light-scattering tissue, such as the brain. Using a microscope to image neural activity has the advantage of being relatively non-invasive. Mounted outside the brain, it simply sends light into it, in turn receiving the light emitted out of the brain. While conventional significant effort over the last years. So far, the state-of-the-art were so-called two-photon fiberscopes that are able of measuring brain activity in freely behaving animals with single-cell resolution. Unfortunately, still many of the cortical layers remained out of reach. With the use of three-photon imaging, one can solve this issue. Due to the light having a longer wavelength, three-photon microscopy is able to penetrate deeper into tissue with reduced out-of-focus excitation. This results in clearer images of structures deep in the Max Planck Institute for the Science of Light. The group of Jason Kerr now developed a small head-mounted three-photon microscope capable of imaging all cortical layers in a freely moving rat. Light is delivered through a custom-designed and manufactured glass fiber. The new microscope allows for continuous imaging of activity from neuronal populations. Even the microscope weighs only 5 grams, a weight well suited to cortical imaging in rats. It is capable of functionally imaging the majority of the cortical thickness of mature rats. The researchers expect the fiberscope to be widely applicable to behavioral research, as previous microscopes were limited in their imaging depth and unsuitable for use over extended periods of time, restricting the types of behavior that could be observed. Researchers are now behavior.
So this is possible. It's possible to measure the network activity
How do
with freely behaving animal. Also optogenetics, I think most of you have already heard about it. It was actually discovered almost 15 years ago, and it's getting more and more common. It's possible to stimulate specific regions, specific neuronal types, which are molecularly, let's say, labeled, and it's possible to stimulate them with light to study their activation and their behavioral outcomes.
Is optogenetics. It all started with algae. In 2002, researchers discovered that proteins are not the only proteins in the that makes this technique so powerful. And channel rhodopsins are not the only proteins in the optogenetics toolkit. In this fly, a different light-sensitive system is used to target just 2 out of 200,000 neurons. These neurons govern an escape response, the reflex that makes the fly flee as you move in to swat it. Here, a flash of light activates the neurons, in a petri to be able to see light-sensitive protein, such as halorhodopsin.
So, what is optogenetics? Basically, scientists use light-sensitive proteins. They infect specific neuron types by viruses which encode for these proteins, and the specific genetically engineered viruses and the transgenic animals make it possible of the expression of these light-sensitive proteins in specific types of neurons. And when you shine blue light on them, you can make these specific neuron types fire. And the network connected to these neurons will, of course, fire as well. And you can observe the behavior of the animal, depending on which animal model you are using. And you can study the connection between network and the behavior of the animal. Lastly, I would like to talk about the single-cell RNA sequencing more, as I briefly mentioned previously. So it's an exciting novel technology. It's basically single-cell omics method, especially single-cell proteomics is not very well developed yet nowadays. but the RNA sequencing and epigenomics is quite developed now and used commonly. It's different from bulk RNA sequencing. It's possible to analyze transcriptome of individual neurons. It's possible to combine different methodologies based on single cell methodology. And it's also possible to study transcriptomics of single cells in spatial resolution. All of these are now becoming quite common. And again, the data sets of all of these omics-based articles are almost always published in open source databases. So you can download virtually any data belonging to an article, and by using, again, freely available Python-based or R-based analysis methods, you can test your own hypothesis without spending any money. It's possible. So in single cell technologies, what is done? First, you get a tissue, in this case, a brain tissue, make it into a cellular suspension, and either by using FACS or by using some specific tools based on microfluidics, individual cells are put into individual wells or droplets, and RNA is extracted, cDNA is constructed, and then this cDNA is sent for sequencing, next generation sequencing, and by using some freely available bioinformatics tools, you can construct t-SNE or UMAP plots, do clustering, do annotation, and analysis depending on your purposes. Again, you can use these methods to study diseases as well, just as we do in our studies. You can compare the healthy brain cells to the, for example, Alzheimer's disease brain cells and detect disease-specific cell types. For example, thanks to this technology, now we know that a group of microglia, which is not normally present in physiological conditions, is formed during disease conditions of neurodegeneration, and this is called disease-associated microglia. Similarly, astrocytes are also changing their phenotype, molecular phenotype, to adapt to the pathological environment, but these cells are also contributing to pathogenesis stem cells because of an aberrant adaptation. By using similar methods, it's possible to study the molecular characteristics of the vulnerable neurons in Alzheimer's disease, because as you know, Alzheimer's disease starts from the entorhinal cortex, which is related to memory function. It's not known why is that so, and it's possible to make studies based on single cell omics methods to study the vulnerability of the specific neurons in this region and the molecular basis of it. As you can see, again, this is a t-SNE plot from by using the combination of neurons from different patients in different disease stages, and each of these dots again are individual cells. It's possible to identify cellular types, oligodendrocyte types, astrocyte types, excitatory neuron, inhibitory neuron cell types, and it's possible to discover novel cell types or molecular basis of diseases. So in the end, although the neuroscience was started with the discovery of the neuron doctrine, we now know that we cannot understand the brain by just studying the neurons themselves. We also must see the brain as a system, as a whole, composed of neurons, astrocytes, microglia, oligodendrocytes, vascular cells, vascular system, all the cellular types working together, and also from different scales, from nanoscale to macro scale. It's a system. So it starts from the molecule and goes to the anatomy. It's a whole complex, multi-level, multi-layered structure, and it's not easy to solve its mysteries. It's nothing that one center or one scientist can do. Even a country like US cannot do it itself, and international collaborations, interdisciplinary projects are now not optional, but it's essential to understand the complex structure and function of brain and also its disorders. So thank you. That's all for my talk, and I can take questions.
Thank you so much for this beautiful speech. Yes, we have a few questions from our participants. Firstly, where are those microscopes that you showed in the slide as the voltage variance and the two photon microscopes? Are they widely available even in Turkey or only in specific research neuroscience labs?
Specifically for two photon, it's available in, as far as I know, in 2 centers in Turkey, although not very heavily used, but they are available. They are so I would like to mention that the technology is not the limiting thing here. So you can just buy it. They are not extremely expensive actually. They can be just purchased and they are available. Almost all of the technologies described here are available in Turkey in at least 1 center, and they are really not so expensive actually. But it's the human source which matters. The human source that can use these technologies, can understand these technologies and can use them. That's what matters.
Okay, thank you. What are the biggest struggles of neuroscientific methods to understand the consciousness, if there is such a thing?
The complexity. So as I mentioned, it's extremely complex. It's the most complex thing in the whole universe. So it's not easy to understand. I'm not even sure if we can fully understand it one time later. I'm not sure about it. It's complexity, and the data amount is incredible. So the data required to fully analyze the molecular structure of the brain from the synapse to the macro scale is incredible. It's said that all of the existing information available in the servers, in the computers, it's even more. So the complexity and the amount of information required to solve brain is incredible, and it's the difficult part.
Okay, thank you. Another question is, what is the exact scope of neuroscience? What methods can we use in order to integrate our understanding of neuroscience to media, especially to cartoons and animation?
I'm not sure if I understand the question right, but animations and cartoons are useful to make things easier to understand, but they also bear the danger to make them also over simplistic. So the video, the media, YouTube channels regarding these journals or institutes are, I think, extremely beneficial, and the Twitter accounts of these organizations are very beneficial. Also, of course, to read the original articles as well, and to seeing these actual data from these large-scale experiments, I think it's very stimulating for us to understand the true complexity of the brain. Of course, it's not. I don't want to give the message that it's too complex, and in Turkey we don't have enough resources to understand this, so we cannot do it. No, not at all. Actually, I'm trying to emphasize the open science concept and the open data concept, because it's really very popular nowadays among the grant organizations. They make it a must. So if you take a grant from ERC or USA NIH government organization, you should publish your data freely in the end. Also, you should develop tools to analyze this data and make it freely available. So actually, every one of our audiences can just download and analyze this. Of course, this requires some effort to learn them, but it's possible.
Okay, and the last question is, what would you suggest medical students in the preclinical years to improve themselves in neuroscience?
Good question, and a topic of a separate meeting, of course. So to follow these institutes. So I would recommend for all of you interested in neuroscience to sign up to the table of contents of Nature and Science journals, because these publications are mostly published there. Of course, when you first read them, it will be extremely difficult, but you should keep on reading because it's only possible to follow the global science by reading it from the first hand. So please freely sign up to the contents of these nature and science journals, and also to the YouTube channels. Just go through them. There are very nice short videos which demonstrate these topics quite clearly. Also, you can use Twitter academically, not to follow up the toxic environment in Turkey, but just to follow up scientists, academicians of your interest, and you can just see a publication as soon as it's published in a journal in just minutes or hours. And they can also frequently they publish tutorials explaining their methodology or their findings. They are all very useful. Of course, I would recommend you to read textbooks if you can find time as well. The Kandas Principles of Neuroscience textbook or Mark Bayer's Basic Neuroscience textbook. They are very useful in the beginning, especially. And also, if you have student clubs, you can do journal clubs among you, and you can discuss articles, because reading and discussing articles is a must. You should be able to search the literature effectively and read and write about the literature. And this can only be done by reading many publications as possible.
Okay, thank you so much for the answers. And again, thank you for accepting our invitation and giving us this beautiful presentation. It was really beneficial.
Thank you.
So our session ends here, and the next session will start at 5:30, so we can have a break right now. Participants can stay on Zoom if they like.